or the taxa that was concerned by the sampling. The findings was really amazing. The first thing we were interested was to assess water properties or water quality inside the forest using water macroinvertebrates, algae, and water properties. So the results have indicated 20 families with key indicators of good water quality, including Chlorosiphidae under odonates, and other groups, 3 families under Trichoptera and 1 family under Ephemeroptera. So this has been concluded based on the sensitivity as it has been developed by the South African scoring system. So the Trichoptera was the good key indicators of water quality with the sensitivity 12. The Trichoptera had 9, and the Ephemeroptera has 9 sensitive scoring system, which is a sign that water quality inside the forest was good. But to conclude about this, we have also assessed the water parameters, focusing on salinity, electrical conductivity, water pH, and redox potential. And results have indicated that the salinity is relatively at low level, and that is a sign that water is not highly saline. And electrical conductivity was also low, which aligns with the low salinity, while the pH was slightly acidic, and this was due to the presence of organic acids, possibly from decaying vegetation, while redox potential was also low, and this suggests a slight reducing environmental due to nutrient availability and water microbial activity. And these findings supports the previous one about water macroinvertebrates, that water quality inside the Mukura Forest is of good quality. This has been also related to water algae because they have been used as bioindicators of water quality in different areas globally. And the findings indicated 10 orders, 15 families, and 25 species, with the key indicators under the family Onotiaceae. And then we have also the Frusturia, with the species Frusturia crassinerivia, and these genus and species are the key indicators of water quality. And we were not able to do the identification to species level of Enotia, which is probably a new species to the area. So we're still working on this. I shall come up with the conclusion after the consultation of experts in algae. So we have also done the assessment of arthropods, focusing on reef litter. And in this regard, we have found 22 orders, 79 families, and the majority of the class Insecta was dominated by the family of Formicidae. But of course, of all sampled arthropods, the class Insecta was also very dominant with 75.7%. We were interested to do the identification of species to species level, and we have found 24 species of butterflies, with the higher number of Nymphalidae with 58.8%, followed by Lycaenidae, Pieridae, and Papilionidae. And we have seen the most abundant in this group being the Pisciclus oliveri and the Caraxes acuminatus, which is a fruit-feeding butterfly. This is a sign that the forest is potential suitable habitat for insects, but due to the shortage of time, we did not be able to do the identification of other families to species level. Of course, there is also limitation of expertise, as we don't have experts which can do the identification of all families to species level. However, the findings communicate the importance of Mukura as a suitable habitat for arthropods and insects in general. We are also interested in the knowledge of epitofauna found in the area, focusing on amphibians and reptiles. One order of amphibian was found with 7 families and 12 species, while reptiles were found one order, the order of Squamata, with 4 families and 5 species. Under reptile, the Cameleonidae family was the most dominant, found in all areas, while in amphibians, the 2 families were the most occurring, Hyperoreidae and Arthropods, compared to other families that have been identified in the area. So the species of Arthropods adolfredelchi was the most distributed, as it was found in 15 sites, but 3 species were least distributed, and these include Phrynobatrachus versicolor and the Phrynobatrachus species, which was not identified, but also the Sclerophyllus chizorewensis, which was found once in one site compared to other 20 sites. And here is we have the photos of some of these species that have been found there. And the mostly distributed under reptiles was the Trioceros johnstonii, which was found in 6 sites. And there was also the least distributed species, which was Ateris nichei, which was found only in 2 sites. The findings have also indicated interesting species of amphibians because the Leptoperis carissimbensis is a vulnerable species that was previously recorded only in Nyung and Volcanoes National Park, and it was found in Mukura for the first time. So there is also another genus, the Phrynobatrachus, which was not identified to species level, and we anticipate to be a new code for Rwanda. We are undergoing more analysis, and we're in touch with experts in the domain to confirm if it will be the new record to Rwanda or if there is something special about this genus. We were also interested in small mammals, where we have found 6 orders, grouping 6 families and 12 species, and they were dominated by the family of the order of Rodentia, with the family of Miridae, where the Lophomoris wasnami was the most abundant species. In this regard, we have found 3 new records in Mukura, which include Heliosirius rufobranchium, and Ilomiscus stera, and Mus bufo. Concerning birds, we have found 124 species of birds distributed into 46 families and 14 orders, where 14 of these species were endemic to the Albertine Rift, and the others were classified depending on IUCN Red List, where we have found one critically endangered species, the Hooded Vulture, and we have also found the endangered species, which compared to other species. I have also greedy blue turaco, which is list of IUCN species. So in general, these species are of great importance because having 124 species within 16 days is a good sign that Mukura is offering a suitable habitat for them, but maybe more and extended research is needed to check if more species were not left behind. We have also sampled and identified the plant species. We have found 255 species, where 245 are native, 10 of them were introduced, and the other 73 were endemic to Albertine Rift. 10 were locally endemic, and a higher number of species were found under 3 families, the family of Rubiaceae, Asteraceae, and Plumeraceae. We were also interested to know about the conservation of Mukura because, as I said in my introduction, the area has undergone different human activities that have disturbed the area. And we have conducted a social science research to identify the participating organs in the conservation. And in this regard, we have found that the Rwanda Development Board, local leaders, communities grouped into cooperatives, security organs, specifically the SOA and police, and the ARCOS now is doing a great job there in terms of promoting biodiversity conservation. And for these people, we have found that the motivation for them to participate include ecological, socioeconomic, and infrastructures. I mean, ecological services, socioeconomic development, and infrastructures, including health centers, market, and honey collection centers. However, the mode of participation is different. Research have indicated that local leaders contribute or participate in community mobilization, in law enforcement, and play the role in the selection of projects to be funded by the revenue sharing programs. But also the community groups are participating in the rehabilitation of degraded areas and planting tree species and awareness raising. However, challenges are still persistent, and some of them are related to human-wildlife conflicts, where the animal called Imhuhwe, which was later identified as hand badger or Merivora capensis, is attacking the beehives for the beekeepers. But there is also a report about the wild dogs which prey on livestock for local peoples. There is also the problem for compensation process that is costly wrong. Here, I mean, the process to get compensated when there is a problem takes a very long process. And the local community members and the other participants in the research was showing that if the channel should be shortened so that it may not take longer time, it may be more helpful for the community. In any case, there is the initiation of compensation process. The other big challenge is the use of Mukura Forest, where there is a frequent entrance of the forest for fodder collection, for timber, for firewood, and for mining, even though the stress related with mining is reduced now. So we came up with some recommendation that participatory governance for effective conservation of Mukura Forest is needed, because these institutions that have been reported, it seems working individually. So there is a need to work in the same forum towards the achievement of effective conservation of Mukura. There is also the need for the development of beekeeping and tourism compared to the neighboring Giskwati forest, where these activities are already developed. Local community members are suggesting that tourism can be developed in the area, and this may include hiking, camping, and this may contribute to revenue sharing. And if you see on the photo, this is an area for camping that has been developed by RDB, by REMA, but it is not working from the time it has been there. And the local community members are saying, if it can be operating, it can be an opportunity for them to get a job and to promote conservation of the area. Then there is a need for alternative source of fodder and firewood, such as agroforestry and planting of fodder grasses and farmlands of local people. The project that is run by ARCOS has started working on this, but the outcome is not yet fully observable so that it may reduce the pressure on the main forest. Another issue is that more research about community development by the University of Mukura are needed for sustainability of the ecosystem goods and services provided by the area. By the forest. So here is the key references that we have been used during this study. Not much studies sure have been conducted here. Most of them were coupled with Mukura, combined the Mukura and the Gishwati, and only one research that has been conducted in 1992 was unique for Mukura. So this study was the first intensive that provided baseline data about Mukura. And of course, we cannot confirm that we have achieved everything. More studies are still needed to supplement the findings of this study. So here we have produced a video that I am not going to play here because it takes some long time. But for those who are interested, I can paste the link in the chat room, and then you can play the video in your free time if you want. So based on the importance of the forest, we came up with our common language when we were doing this research that Mukura is our forest, but it is also our life. I thank you very much for your kind attention, and if there is a question or any intervention, you are welcome. Thank you very much. She might be muted, maybe.
Oh, good evening. Actually, it's already good evening.
Yeah, depending on where you are.
So much. Yeah, it's already. We are home. I know it's already good evening.
Okay,
thank you, the speaker, for the informative presentation. It's really very interesting. Yeah, in fact, it's not that much a question. It's like acknowledgement, if I can say like that. So first of all, if it's possible, maybe to share the presentations. Actually, I could not really, I was enjoying the presentation, but I could not really follow because of the difficulties that we encountered. So please, if it's possible, you can share us the presentation as well as the link for the video you mentioned. I think that one it's already there in the
Yeah,
chat.
there is no problem. At the end of this presentation, I can share it. I will send it to Vena, and Vena can share it to anyone who want to explore more.
All right. I'll finish there. All right. Thank you.
Good. Thank you, Vena.
Actually, again, I wanted to find out there is a local NGO that's always working around the Forest of Hope. So when you in your last presentation you mentioned about the participating organs. So I didn't really see this one. Is he no longer working? The Forest of Hope. I know they are working in the Bos, Jishwati, and the Mukura, and it's a key. It has started some of the tourism activities within the two forests, which are now national parks. I don't know if it was not among the participants in your group discussions.
Yeah, thank you very much. We have seen that actually when you talk about Gishwati Mukura, that is one national park. But the two forests are far separate. There is around 50 kilometers from Gishwati to Mukura. So most of the activities are taking place in Gishwati, but Mukura seems as left behind. So the tourism activities, the Forest of Hope activities are very active in Gishwati. But in Mukura, sure, there is only RDB with park rangers doing a great job there. And around the forest, there is a big project of ours called the Murachira that is doing agroforestry with the local community members. But concerning the conservation of Mukura, for sure, no tourism activities, no community activities developed like Gishwati. Actually, even though they are making the same park, but they are not managed in the same way in terms of tourism and in terms of conservation of the area. So Forest of Hope, I know it. It is very active at Gishwati, but not very, very active in Mukura compared with the achievements they have in Gishwati. Thank you.
Yeah, all right. That was all about the comment. Thank you so much.
And in addition, this is also related to the bioassessment. You will find more publications about Gishwati. When you go in the topic will be Gishwati Mukura, but when you assess the methodology, you will find that the study has been conducted in Gishwati only. Mukura is not concerned. So the two references I have showed you at the end, they're the only studies that have been conducted in Gishwati, Mukura, and as I said it, only one has been conducted only for Mukura in 1992. Others combined Gishwati and Mukura, and there are more studies in Gishwati compared to the studies that have been conducted in Mukura. Thank you.
Okay, thank you very much, Professor, for your nice presentation. Some of us, we try to remind us how Mukura is, specifically for their biodiversity. But I would like to know whether among of introduced species, 16 species that have been presented, if there are still some exotic like character species, because there was a project which was intended to remove those specifically those exotic species. I would like to know whether those exotic species they are still there. If yes, what should be like the measures that should be taken? Because different research which have been done showing that those exotic species they have some negative effect to biodiversity species. Thank you.
Thank you very much. There are some traces of eucalyptus at the edge of the park, but at a small scale. They are not really much. And our study was not interested with the area concerned by exotic species. We were interested with the native forest, inside of the forest. So for those remaining exotic species, what can be done is continue to remove them until the forest it will be only dominated by native species. However, there is also a need of a buffer zone for this area. As you know, you get out of the forest, you are already in the lands of the people. And if you remove those eucalyptus, then you immediately enter in the forest. The good thing is that they are not on a big scale. There are some traces because, as you said, there has been a project that has removed them. I think in 2005, if I'm not mistaken, something like that. I don't remember the time very well. Thank you. I don't know if I reply positively to your question, but that solves this situation. There are some traces of eucalyptus.
Okay. Okay, thank you very much. It was 2015. Yeah, thank you.
Yeah, 2015. Thank you.
Mm-hmm. Yeah, that's so. Even the paper is online. Those who are interested, they can read. Yeah, yeah. Mm, mm, mm.
Okay, thank you, Vena, for the supplement.
Good afternoon, everyone.
Good afternoon.
Thank you so much, Mr. Vena, and the presenter and my colleagues here. My name is Emmanuel Ntawizigira. I would like to thank the presenter for this wonderful presentation. It was insightful, informative, even inspiring for us young researchers who are still on the way of development. So I think I have like two questions.
Yes, please.
Number one is about it's on a title, Biodiversity Data in Mukura. So I think according to what the presenter has presented, it is all about animal biodiversity. My first question is that is there any maybe the next seminar if we you could also bring a second session talking about or presenting about plant biodiversity. I was once in that forest. I was collecting data. I think it was last year, like in November or October. So, but because I had a short time, I didn't spend there a long time. So, my suggestion is that if you can also bring the data about the plant biodiversity, it will be completely informative. I think I would like to know if there is also another presentation talking about the plant biodiversity. The second question is about I would like to know maybe if you can give us some insight like about the threats. I have seen recommendations, but I don't know if maybe I was disturbed by network, but maybe you can share with us if biodiversity you have presented to us. Thank you so much.
Yeah, I think I have done the presentation about plants. If you look at the screen, there is at 7, it's about plant diversity. But of course, this is a summary. We have a detailed report. Maybe for those interested to know more about the plants diversity, we can share the report. But we have talked about plants as you see on this shared screen. Can you see the screen?
Yes, please.
Yeah, so we have talked about it, and our research was also about plants, and we have got good results. Concerning the threats, we have also talked about them. If we go on this slide about challenges, and among them there are some threats that I've talked about in the previous in the introduction, and those threats, some of them are still existing in the area, such as encroachment, local communities entering the forest for looking for food and firewood, some illegal mining activities, they are still there. But for the mining, maybe the frequency is reduced, but some traces are still there. I can remember even by the time we were sampling, there has been some kind of conflicts with the park rangers. So the challenges or the threats are still there, and some of them are detailed here. And the reference to your first question, we have also sampled the plants, and maybe I will share with you the PowerPoint as Andrew suggested it, and you can see more details. I can also give you the full report that you can read, but even though it is not published, but you can get more details about it. Thank you. I can see Adrian asked about the number of species of butterflies. Well, find 24 species of butterflies, and the Nymphalidae family is the most dominant. If you're interested also, I can share with you the detailed list of the butterflies you have identified there. Divine? I think Divine has also raised a hand.
Thank you very much. I really appreciate or grateful for this interesting presentation. I would like to ask on my side about macroinvertebrates. Mukura and Gishwati are on the same park, and I would like to ask if you did the comparison between macroinvertebrate on Mukura and the Gishwati, and if the macroinvertebrates are the same. Thank you.
Great. Thank you very much. This research was specifically for Mukura. I know there is some data collected from Giswati, but I didn't want to do the comparative study because they were not sampled in the same time. They were not sampled by the same people, and they were not, sampled in the same time. And for macroinvertebrates, to conclude and bring in the comparison is that if it is the rainy season, you do the sampling in the rainy season, you use the same sampling methods, and you use the same identification process. So the data I presented here are only for Mukura, and I did not do the comparative study because the data from Mukura, I don't know how they have been sampled. I don't know how they have been analyzed. But anyway, I have the report there. I can check and see. But the data presented here are only from Mukura. Thank you.
Yeah, sure. Thank you. Thank you.
Thank you very much. Bye. Have a good time. Bye.